So first of all, thank you for inviting me here to give a presentation. So I'm not a data scientist. This is my official disclosure, but I am generating data. And we use omics technologies to do that. And what I thought would be a good exchange of ideas is what are the caveats and what are the opportunities of generating this new data, and what do we need to look out for if we want to apply it to big data, because whatever we are going to measure, it's going to be expensive. So we better make sure that it's meaningful and it actually really advances medicine. So I think here's where we are at the moment. We have a wealth of genetic data, but in the end, we use genetic information as a proxy for protein function, because the molecules of life are not the genes, it's the protein, it's the molecular machinery that actually mediates disease. The genes are just the blueprint. So the question is, to what extent do we need to complement the genetic information with additional protein information to get more out of the wealth of genetic information that is already out there? And I always show this slide to my students, but I actually think it's very useful. So the genome, in principle, is what might happen. The transcriptome, in principle, shows you what is likely to happen, but it's also very temporary. Transcripts are only temporarily expressed, so a transcriptomic profile is very variable compared to a protein profile. Proteins, on the other hand, is showing you what is actually happening, and the metabolome shows you what has happened. Now, what we have available in large cohorts is genetic information, and to some extent now metabolomic information. We don't have much in between. We don't have much information on RNA data or protein data, and in particular on protein data, because although the human genome has 20,000 protein coding genes, there are estimates that we may have well over 200,000, if not 2 million different proteins. So the biological complexity is not determined by the number of genes we have. In fact, a simple rice plant has 40,000 protein coding genes. So a simple rice plant has twice as many genes as a human being, which tells you that biological complexity is not mediated by the number of protein coding genes you have. It's what you do with this set of genes, and that is determined where you are at the protein level. So the challenge in having protein information is that we do not measure proteins directly. If we currently measure proteins, we usually use antibodies or aptamers. So we rely on binders to measure the protein. And there are two major caveats. First of all, we don't have good binders for many of the proteins we would like to study. And second, you can have a good binder and a bad binder, and your information and your outcome may not be determined by the protein you're measuring, but the quality of the assay you have to measure that protein. So a good biomarker may make it through as the gold standard biomarker because it has a good assay, not necessarily because it was the right protein. And mass spectrometry, of course, gets rid of that because we measure all the proteins on the same machine in the same run, so we can compare like with like, rather than comparing an assay from one vendor with an assay of another vendor. Having said that, there's a wealth of mass spectrometry data out there, and this is actually deposited now in public data repository, and I think our group contributed the most entries on cardiovascular disease to date. But if we look at all the mass spectrometry data that is out there, we still have about 2,000 proteins that we know are encoded in the genome, but for which we do not have experimental evidence that they actually exist. So the protein products of about 18,000 of these genes have been detected by now, at least in one of the human tissues, but 2,000 proteins are still missing. And of course, we have a map of the human genome, but we do not have a map of the human proteome. And I think this is one of the challenges for the GWAS studies. You get a hit for a certain gene, but the next question is, how is that gene actually mediating its function? And the first question is, where is it actually expressed? And where does the benefit or the detrimental effect come from? So most successes in cardiovascular disease were for genes in lipid metabolism because you knew you need to look in the liver. But with other major hits we got from GWAS, we didn't make much in vain in order to understand how they actually link to the pathological mechanism. And much has to do that we don't even know which proteins are expressed in which tissues. Now, with regards to the human plasma proteome, we face a particular challenge, and this is that the human plasma proteome is the most complex proteome of the human body. You have proteins like albumin that are present in the milligram per milliliter range, and you have, on the other hand, interleukins and cytokines that are present in the picogram per milliliter range. So you have a dynamic range of 11 orders of magnitude, and there is currently no single technology that can measure those proteins across the entire range. What is happening is that mass spectrometry, for example, we now measure proteins down to the nanogram per milliliter, whereas we then use, for example, proximity extension assays to measure proteins in the picogram per milliliter range. So by combining different omics technologies, we can span the entire range, but there's not one solution for the entire protein range, and I think this is important to keep in mind. One particular area is highlighted here in blue. These are proteins that leak out of diseased tissues. And I think this is, of course, where one of the most famous biomarkers in cardiac disease came from, and this is cardiac troponin. So cardiac troponin is a cardiac-specific protein. If you have a heart attack, your muscle dies, and this protein, which normally isn't detectable in healthy individuals, actually is detectable in patients with heart attacks. Having said that, this protein biomarker is one of the most successful ones because it is so specific. It's specific for the heart. But we did actually a proteomic study together with Professor Manuel Mayr, and we actually found that there's another cardiac-specific protein called cardiac myosin-binding protein C that shows a more favorable release kinetics compared to troponins. It actually performs in clinical studies similar to troponin as a rule-in marker, but it performs better as a rule-out marker. So you actually could potentially reduce hospitalization time because you have got an additional biomarker for cardiac injury that shows a different kinetics compared to troponins. But I would like to highlight this not just because of a translational success, how proteomics led to a potential new biomarker candidate that's now licensed, but also to highlight the fact that there are surprisingly few tissue-specific proteins. For cardiac-specific proteins, we probably have only got about 5 cardiac-specific proteins that are not expressed in other tissues. So what this tells you is that big data then often isn't necessarily the solution. So these are data from our colleagues in Harvard, where they also looked for biomarkers of cardiac injury, and they used an Aptamer approach. So in the first phase, they looked for 1,000 proteins, and the top candidate was troponin. In the second paper, they looked for 5,000 proteins, and the top candidate was troponin. Why? Because cardiac myosin-binding protein isn't part of the Aptamer panel. So you measure what you can measure because you're given a set of tools, a set of binders, and this is what you're measuring, and this is the answer you're getting. But you may miss important information because you don't have an unbiased technology where you look for all the proteins that you actually can measure. And also the homologic scan is very widely used. I think this is based on nucleotides which bind to proteins. There are also several issues, in particular with cross-reactivity and unspecificity. So I think this is just another caveat I want to measure. There's commercial solutions out there. Some of these aptamers may be very good and very, very trustworthy, but the problem is you don't know which one of them is actually giving you the right protein information. And what you don't want to map is a genetics map for protein concentrations if you actually have a genetics map for aptamer binding, because the genetic variation may determine the binding of the aptamer. So I do think there's a need that we measure proteins directly, and then of course we also need to use these measurements in clinical cohorts. But again, I think this was already mentioned before. The major issue is confounding, and we don't want to just compare cases against controls. We actually want to have a longitudinal measurement where we can take repeated sampling in the same individuals over time so that we can determine when the biomarker changes in relation to the onset of the disease. And this is something we have done extensively with one study in Bruneck in southern Tyrol. So the major advantage of this study is that these individuals were followed up over 25 years. They have 5,000 clinical data points per patient. And we have added 500 molecular markers, mass spectrometry-based proteomics and mass spectrometry-based lipidomics, including some RNA species. And there are several publications that have emerged from this work already, but I wanted to show you one particular example which relates to one of the major risk factors for cardiovascular disease, and this is lipids. Because obviously cardiovascular disease manifests itself usually in our 50s and 60s, but actually it already starts from childhood. And the reason is that we have lipids in our circulation. Before and after a meal, you can see this change in turbidity of your plasma. These are your circulating lipids. And unfortunately, some of the lipids over time will make it into your blood vessels and end up as fatty deposits in your arteries. So rather than just looking for the endpoint, the heart attack and the stroke, what I want to see is how actually the disease progressed in the individual over time. So the most interesting association is not the endpoint. The most interesting association is actually the progression of the disease over time, and this has to be monitored over decades. So what additional information can we get, and what are we measuring right now? And this is a quick introduction to lipoproteins. So lipoproteins are basically an envelope for the lipids that are not water soluble, and they are carried in these lipoprotein particles. They have a lipid envelope and a protein that's wrapped around it, and these proteins are called apolipoproteins. And if we talk about big data, then this is what we currently do for cardiovascular risk prediction, and I think we can all agree that this is anything but big data. But it is very cost effective to measure. We record the age of the patient, we look for a family history, we ask them whether they're smokers or non-smokers, whether they have diabetes or not diabetes, measure their blood pressure, and then in principle we only have 3 lipid variables. And this is LDL, the low-density lipoprotein, which is the bad cholesterol, the high-density lipoprotein, which is the good cholesterol, and then triglycerides, but triglycerides at this point in time don't really inform treatment decisions. So it's a very simplistic set of measurements, given the fact that we know cardiovascular disease is caused by lipid abnormalities to a large extent. And since I've graduated from medical school, none of these measurements has really changed. So if we say that the genetic risk score improves risk prediction, we have to bear in mind what we currently use for risk prediction. The bar is relatively low. Having said that, I think it's also interesting to note that in many studies we don't measure LDL cholesterol. Also, LDL cholesterol is the major risk factor for cardiovascular disease. It is often not measured directly. The reason is because it's more difficult to measure. So it's easier to measure total cholesterol and HDL cholesterol. So in many of our clinical studies, we just calculate LDL cholesterol by subtracting HDL cholesterol from total cholesterol and adjusting for triglycerides. And this goes on when we then look for protein measurements, because there are many apolipoproteins encoded in the human genome, but if you look at the cardiovascular literature, in principle, we only measure 2. ApoA1, which is the main apolipoprotein on HDL, the good cholesterol, and ApoB, which is the only apolipoprotein on LDL, the bad cholesterol. There wasn't any single study that actually has compared the different apolipoproteins against each other. So this is what we set out to do. So as part of our mass spectrometry data set, where we measure about 150, 200 proteins, and soon we're going to be able to measure 300 or 400 proteins in the same run, we actually looked just at the apolipoproteins, and this is based on 688 individuals. We measure 13 apolipoproteins, and rather than measuring HDL, we now measure the subset of apolipoproteins that associate with HDL and that determine its function. And rather than just measuring ApoB, we now actually split up the ApoB that's associated with LDL, and importantly, we separate it from another group of lipoproteins that are called very low-density lipoproteins that also carry ApoB. And we can see these distinct clusters, and we want to test the hypothesis whether indeed ApoB and ApoA1 are the 2 best apolipoproteins we can use for cardiovascular risk prediction. And the answer is here. So this is in this PRONEC cohort. There were 100 events over 10 years and about 600 controls. And these are the associations of these apolipoproteins on the y-axis with the incidence for cardiovascular events on the x-axis. You've got the hazard ratio. We didn't see a pronounced reduction of cardiovascular risk with ApoA1, but of course, as expected, you did see a very pronounced effect of ApoB100, so approximately a 30% increase in your hazard ratio for every 1 standard deviation higher concentration of ApoB100 in your blood. But if you actually compare the levels you get for ApoB100, you can see that the associations for apolipoprotein E, ApoC3, and ApoC2 are very similar to ApoB. In fact, some are even stronger, and if you use any one of those measurements and adjust apolipoprotein B100 measurements for apolipoprotein E, C3, and C2, then ApoB100 loses its significance. And what does this tell us? Well, it tells us that there is cardiovascular risk beyond LDL cholesterol. LDL cholesterol only carries ApoB100, but ApoB100 is also present on very low density lipoproteins, and the other apolipoproteins I've just shown you, ApoC2, C3, and ApoE, are not found on LDL, they're only found on VLDL. So again, I think there is here this dilemma that certain things we are measuring in the clinic may not necessarily be the most informative ones, and in the era where we now lower cholesterol very efficiently, there may be a certain residual cardiovascular risk that is actually captured by other apolipoproteins, which we may want to include in measurements, at least in clinical studies and clinical trials for medication. So to paraphrase Stephen Hawking, the cost of poor data is the illusion of knowledge. If we measure ApoB, we are not measuring LDL. We are measuring all the lipoproteins that carry ApoB, and this is VLDL, IDL, and LDL. Similarly, if we measure HDL or ApoA1 lipoproteins, we have a different class of HDL. We've got small, large, and very large HDL particles, and they do not carry the same cardiovascular risk. So we actually need to break down these lipoproteins into these different subfractions. We did it by mass spectrometry-based proteomics. You can also do this by nuclear magnetic resonance spectroscopy, where you actually look at the different lipoprotein particle sizes. And this was done by a group in Oxford in the China Kadoorie Biobank, and Michael Holmes is here the first author. And it nicely complements what I've just shown you on the apolipoprotein side. So our data was proteomics-based. This is now metabolomics-based. You use an NMR spectroscopy instrument to resolve these different lipoprotein particles. And the first thing you realize is that actually the cardiovascular risk, again, is not only associated with the LDL particle. You get a similar cardiovascular risk associated with the VLDL and the IDL particles, which predominantly carry triglycerides. And for HDL, you see that the large and medium-sized HDL particles are the ones that are protective. Their cholesterol content is associated with a reduction in the hazard ratio for myocardial infarction and ischemic stroke, whereas actually the very small HDL particles are neutral or even detrimental. And also cholesterol is a very promising and a very proven treatment target for cardiovascular disease. We now live in an era where actually we give statins to our patients, we lower cholesterol, and the triglyceride association you see here with myocardial infarction and ischemic stroke may become more important in an era where one of the major risk factors, cholesterol, is so well controlled for by giving medication. So this is now a new hypothesis that it's not just LDL cholesterol that actually mediates atherosclerosis. It's also these remnant particles of triglyceride-rich lipoproteins that actually can cross into the arterial wall and actually mediate cardiovascular disease. And this is supported by genetic evidence. So there were loss of function mutations in this protein called ApoC3. And as we mentioned before, these are experiments of nature, but they are very rare. So in this case, they had almost 75,000 non-carriers and only 260 individuals that were heterozygotes. But these individuals have a reduction in their triglyceride levels by 44%. ApoC3 is one of these inhibitors of a lipoprotein lipase. This is the enzyme that clears your lipids after your meal. And ApoC3 is the inhibitor of this enzyme. So if you have a loss of function of this inhibitor, you get faster lipid clearance. And by reducing your triglycerides, not only do you reduce your lipid levels, you actually also get a reduction in coronary disease. So again, of course, this is now some evidence that actually lowering triglycerides may be an additional therapeutic option, in addition to, of course, the proven effect of cholesterol-lowering medication. So I think the key point I would like to get across is that cardiovascular diseases are not purely genetic diseases. There's a genetic predisposition, but there's a big environmental factor. And we heard about Harry talking, about the stairs and the elevator. Well, if you actually do physical exercise, your genetic predisposition doesn't change, but clearly your lipid levels and so on respond. So you need to take these environmental factors into account. And I think this can be done by not just focusing on the genome, but by adding additional measurements such as protein and lipidome measurements to the genetic information we have. So to combat this mismatch between the wealth of genetic information and the paucity of molecular information at the protein and metabolite level. So I think this is my conclusion slide. I think big data are important, but equally it's important that we have the right data. It can be very useful to have structured data because we don't know where the proteins are coming from and what determines your levels. So for example, most biomarkers in the cardiovascular literature often haven't been controlled for by the confounding of heparin, because heparin is what we give our patients if they are undergoing a cardiovascular intervention. So often you get biomarkers that are triggered by heparin rather than by the disease itself. And the other problem, of course, we need to do this measurement at scale, and it's often not the researchers who do these measurements anymore, but we are outsourcing these measurements. And the agenda of a commercial company is to offer you many measurements for a small amount of money. So often there's a bit of overselling going on in what they can measure compared to what they are actually measuring. And I think we have to address these methodological issues and not simply rely on outsourcing and on companies to provide us with the correct measurements. And having said that, generating data isn't enough anymore. So we used to be all experimental biologists in my group. Now 1/4 of the group is looking at bioinformatics, and I think this is clearly the next bottleneck. We can measure 500 proteins in now thousands of samples, but what are you going to do with this information, and how can you deal with the data in a more comprehensive way? And Dr. Konstantinos Theophilatis, who is here, is heading a small team of bioinformaticians, and I think this initiative is very timely because we clearly need this support for data analysis as much as we need new technologies to actually measure proteins reliably. And with this, I'd like to end and thank the British Heart Foundation for my funding. You can still use this link with the EU domain for the next 3 months, but I've already been sent the official email that after Brexit this will be not functional anymore.
Thank you. Thank you.